Our next speaker is Gabriel Zayas. He's a postdoctoral research associate in the Department of Animal and Dairy Sciences at UW-Madison, working in Francisco Peñagaricano's lab to apply quantitative genetics and genomics to improve understanding of complex traits in dairy system. Let's welcome Gabriel to the stage.
Hello everyone, thank you for coming. Today we're going to talk a little bit about heat stress and a little bit about epigenetics. Okay, so let's start with the heat stress just because it's sort of a little bit simpler, but we know a lot about heat stress in dairy, right? There's always going to be this immediate effect on lactation in your lactating cows that will result in them eating less because of that heat stress. That then leads to lower milk yields. You also have issues with fertility and then some health issues. But these are quite easy to identify, right? They're short term, right? Some of them are a little bit harder to actually pinpoint, right? And those are going to be some of what we call these delayed effects in the non-lactating part of the herd, right? In a sense, heat stress can program animals at an individual level, and you don't see that reduction in productivity until many, many months or weeks or in our case, generations later. Right? So we're here looking at epigenetics and its relationship to a multi-generational decrease in production because of heat stress, right? So specifically what we have here is a dry cow that is pregnant, right? Here you have the fetus, and so this mother is what we call the F0. If we compare a mother that experiences that heat stress versus one that has a heat abatement, right, it's allowed to cool off, we see that those that experience the heat stress do tend to actually have a lower in milk production. In addition to that, we see that fetus that is experiencing that heat stress at this specific event does, when they are developing, have stunted growth, stunted mammary gland development, issues with immunity, survivability, a bunch of different things, right? And it makes sense logically, right? That animal is developing in the natal environment and is exposed to this chronic heat stress that will cause a lot of different issues. Interestingly, the great-granddaughter of that F0, the F2, also has some issues, and it's technically present during this heat stress event in that F1 daughter, right, as an egg. And so we see that even this F2 has issues with early culling, reduced production, and once again, that stunted mammary gland, right? So we're seeing across 3 generations that were present during that heat stress event, a lowering in production and also welfare of these animals. Interestingly, and this is more new data, we're seeing that the F3, which are the daughters of the F2 that have never had that heat stress event, were never present, are also having issues specifically with their mammary gland development. And that's what you can see here in this figure right here. On the left is the great-granddaughter from a heat stress, and then on the right is one that was cooled, from the same event, right? And what we find is that some of the non-luminal epithelial structures are different, right? And it all sort of points to a lag in development in these heat stress individuals. In addition, we see that things like in the heat stress, there's a lower cell proliferation occurring, higher cell turnover, right? And both of these phenotypes, we already saw them actually in the F1 and the F2, right? And we believe it's what actually led to that reduced production. And while these individuals haven't lactated yet, we are assuming, or we hypothesize, right, that they may also have a lower production. And so the real question is, why are we seeing this, right? The F3 is never there during that heat stress event, and we are wondering why, and it's our hypothesis that epigenetics is the reason for that. Epigenetics is the study of how gene expression changes without actually alternating the DNA sequence at the nucleotide level, right? And there exists a bunch of different mechanisms by which that happens, right? In our case, we're going to be looking at this specific one called DNA methylation. And just very simply, if you go back to when you took biology or genetics, remember that the DNA has 4 different nucleotides, right? And those are all 4 different bases. And so essentially what methylation is that we exchange this hydrogen right here for a methyl group, right? And the actual outcome of that is actually very sensitive or very dependent on where that methylation is occurring in the genome. If it occurs in regions like the promoter region, it can actually curb gene expression. If it's in the gene body, it can influence the isoforms that are being expressed. So it's very context specific, but we know it's very important for how an animal regulates its gene expression. So we hypothesize that that heat stress event induced DNA methylation changes that are contributing to that mammary gland phenotype, that those effects are persistent across generations, which is why we see that phenotype reoccur every generation. So our objective is to, first of all, identify what we call differentially methylated cytosines in the F1, F2, and F3. So these are the different loci that are being methylated. We want to then identify which of those methylation patterns are in common across generation, annotate those to the different genes that are present, and see what are the biological pathways those genes are important for. And so here we have the experimental design, and like I said, the original design, so this is the F0, it was actually originally in Florida. We have our heat stress group here, and then we have our cooled group here, and we actually track their descendants for the F1, F2, and F3, and all of these animals are sort of in the same contemporary, right? So after this treatment, they're generally experiencing the same sort of environment compared to each other. We then, at weaning, collect mammary gland tissue and actually do whole genome bisulfite sequence. We process that with bioinformatics, and we do our different analysis, okay? And what we have here, first of all, are the differentially methylated cytosines that we have each generation. In blue is going to be hypomethylation. That means in the cold group, it is more methylated than in the heat stress group. And in red is your hypermethylation, which is just the opposite. More in heat stress, less in the cooled. And these are the results we get. So on the top is your F1, F2, and then your F3. And as you can see, there's a huge difference as you go from that initial heat stress event, each generation, where as you go to the F1 to F2, it goes about down by half, and then F2 to F3 as well. So these are all going to be methylations that are in that small to moderate effect size, right? When we look at larger effect sizes, it's actually quite similar. As you go through each generation, there is that reduction. We see that the F1 has 2.1 times as many DMCs as the F2. The F1 has 7.5 times as much as the F3, and then the F2 has 3.6 times as many as the F3, sorry. So, while that being said, we have millions, hundreds of thousands different methylated cytosines, but when you look at the overlap, it's actually quite few. So here we have the Venn diagrams for each of those, and so there's about 352 that are in common between the F1, F2, and F3. For hypomethylation, there's only 183. So we have a lot of different methylated sites within generation, but they're not always transferable. That being said, if you actually look at the expected by chance in common, this is actually about 12 times as many as they're supposed to be just by chance. So it's definitely enriched slightly, but it's definitely not a lot. But when we actually annotate these DMCs to the actual genes, we have a much clearer story. What we find is that, first of all, the F1 and the F2 have about the same number of genes here, while the F3 has only about 5,500, the other two have about 13,000. And when you look at that overlap, we have a much clearer signal, more coherent. That F3, almost all the genes that are differentially methylated are part of this three-way overlap. So what this tells us is that while the DMC unit is not generally the same, the genes that they're targeting seem to be exactly the same, at least between the F1, F2, and F3. Obviously, there are some that are unique to the F1 and F2, and unique to the F1 and unique to the F2. Now, what's even more interesting is that if we look at the next level, right, which is, genes are genes, but genes have a function biologically, right? So if we look at the pathway level, we see once again a very clear image. So on the left here, we have DMGs that were annotated from the unique DMCs from F1, F2, and F3. On the far right are going to be those DMCs, those 353 and 183 DMCs that were overlapping across all three. On the left here, we have different biological pathways, right? And this is just a heat map where the darker it is, the more significant that pathway was enriched in our analysis. And so what we find is, first of all, there's this very clear Francisco's likes to say this is my favorite pathway, and it sort of is my favorite pathway because it explains a lot of what we're seeing in these other downstream pathways. The first few being about signaling that are very important, different signaling pathways important in the mammary gland. But then about in the bottom here, we see a lot of cell adhesion molecule interactions, gap junctions, things that connect cells together and are important for tissue formation. And what we find is that this sort of upper level IGSF CAM signaling is first of all categorized as an environmental information processing pathway. So it recognizes the environment, such as heat stress, and then also it signals molecules downstream, which is exactly what we're seeing here in these pathways and are very important for normal mammary gland development. So what we're seeing is that DMCs are sparse in common, the DMGs are more in common, but the biology all points towards the same direction. Now, methylation doesn't necessarily mean that it's going to be expressed differently. So what we did is we actually looked at this pathway in the context of gene expression using RNA-seq, and in this case, we had data from the F1 at birth and at weaning. And so we conducted what is called a gene set enrichment analysis. It's to see whether the genes that are in this pathway are more likely or more often up or down regulated. And that's what this plot sort of shows. So this x-axis is going to be, right here you have all your genes, and those that are highlighted are the ones that are part of our pathway. If you're on the right, you're down regulated. If you're on the left, you're up regulated. And this plot right here, these lines just show the enrichment of this pathway for these genes in birth and then for weaning. And what we find is that this pathway is significantly enriched for down regulation, meaning a lot of these genes at birth and at weaning are being down regulated within within our samples, right, in the heat stress. So just overall, some key takeaways. We see that the methylation at the cytosine level is weakening across each generation, right? However, what we see is that there is a conserved course of DMC and more importantly, those DMGs that are persisting into that last F3 generation. We see that this important pathway is significant for both methylation and gene expression, and that in general, it's being downregulated at birth and at weaning and might be driving this mammary gland phenotype we're observing. In the future, we're actually going to try to identify or pinpoint specific DMCs or DMGs that are driving these changes in methylation expression and ultimately our phenotype. And then we actually want to establish that different methylation within the germ line to establish a transgenerational inheritance. These mammary glands are somatic cells, right? Somatic cells aren't inherited, but germline are. So if we can identify those candidates within the germline, that means they are being inherited, which sort of follows what we're seeing in the mammary gland. And lastly, if we can identify those, we can find ways to hopefully mitigate it or try to reverse it. I'm not an expert in that, but I do know you can do things like providing feed additives to change methylation signatures. And then just in conclusion, heat stress in the great-grandmother has outcomes on that great-granddaughter, and it seems that a lot of it is due to that DNA methylation. We also see that we have that phenotype being persistent across each generation. And while the F3, we don't know if they have milk production reduction, it seems it does. But this has very important implications for things like production, but also animal health and welfare. And as the climate is changing, right, these might become more prevalent over time. And yeah, I just like to thank the funding sources, the Dairy Innovation Hub, the USDA, La Porta, because this has been, maybe a decade or more of research. I'm not really sure. And I just got here, so yeah, I thank all of them. I don't know who they are at this moment, but I do thank them.
All right. Thanks, Gabriel. Questions?
Fascinating research.
Fascinating research, huh, Amanda? Wow, where did you get those DNA samples?
Sorry, what?
Just kidding.
Okay, I was like...
So I guess I have a question. Mm-hmm. We always pay attention to the daughters. If you have heat stress bull calves, has anybody ever shown that this methylation pattern can be transmitted to the next generation through the male side? So
for sure, there's research that just, in general, does show that the males through the sperm do actually transfer methylation. For this specific experiment, they looked at, I believe, methylation in the liver of the bull males, and they did find, due to that heat stress event, differences in methylation. I believe that was the F1. But yeah, you do see it, yeah.
Other questions?
fantastic presentation, Gabriel. I have a little question about the number of genes that are affected each generation. So there are, the first F1 generation, there are 13,500 something genes, which is almost like 65% of the protein coding genes, right, of the cattle. Mm-hmm. How do you explain, and the next generation around like 10,000, how do you see heat stress as an environmental factor can affect the regulation of 13,000, 10,000 genes across generations?
So I will just note, those are not all protein coding genes. Some of those are like non-coding RNA or other non-protein coding. So the space of protein coding is about 25,000. In our case, I think it was like 40,000 in total, right? So that's why that number is maybe a little bit higher than you'd expect. But I would expect methylation in a heat stress event to have changes in many genes. Whether they're all important is an important question, right? The reality is that methylation doesn't necessarily just change gene expression, it changes alternate splicing. And you can also have methylation that maybe doesn't hold a function in that specific time point. Remember, we're taking a snapshot of what that genome looks like, but it could be that methylation marker hasn't gotten rid of yet, hasn't been rid of yet, and had an important factor beforehand, right, rather than in that specific moment, right? Methylation changes basically the gene expression of every tissue, right? And we're looking at a bulk bulk whole genome bisulfite sequencing, so we're looking at maybe cell-specific methylation patterns as well as just general methylation patterns. But yeah. You're right. Yeah.
Okay, Victor.
Thanks, Gabriel. Very, very, very interesting. So just a curiosity, I guess. So you're following F0 through generations, and I guess you're controlling that F1, F2 don't have heat stress. F0 was the one with heat stress. What do you think it would happen if there is heat stress across?
So I will say, I can't tell you that those F1, F2, F3 never experience heat stress. What I can tell you is that also those cooled would have experienced that same heat stress, right? They were not individually housed, right? The only treatment level that was given, that heat abatement versus heat stress, is at that mother's level. Now downstream from that, they're all sharing the same environment, right? And so if they're getting heat stress, they're all getting heat stress at the same time, right? That being said, we do see huge differences, not just in the methylation, in the gene expression, but at the tissue level, right? So there's definitely something biologically important that is occurring due to that heat stress event, despite, you know, having, I'll guess, more noise because we're not controlling for that environment, but it's also going to be very difficult to control for environments like that. Yeah. Hopefully that answers.
Okay, let's give Gabriel another big round of applause.